Okay,
Yes.
so today's talk is more of like interactive session, as Dr. Gopal ji has mentioned that this is like informal kind of chat series where we learn a lot after listening to the speakers. And my purpose here is to bring a topic which can be understood by bachelor's degree students as well as postdoctoral scientists or young faculty members. So today I'm going to discuss a very important topic that is known as induced pluripotent stem cells, which has lots of potential, and also 3D culture system, which is an emerging field in the regenerative medicine, and how these iPSCs and 3D culture systems are transforming regenerative medicine and drug discovery. So before I go into deep of my presentation, I would like to give an overview of the talk. So first, I will discuss about different phases of the drug discovery, because everything, as you can see from the topic, that everything is related to drug discovery. So before understanding how iPSCs and 3D culture systems fit into that topic, it will be a great idea to introduce you, all of you, to the different phases of drug discovery. And then I will come to iPSCs, what they are, how they were discovered, and who got the Nobel Prize, and what was the significance of these cells. Then I will come to 3D culture system, which is basically spheroids, organoids, and embryonic bodies. And then their applications in regenerative medicine and drug discovery. What are the key challenges and future perspectives? As most of us know, as most of us know, hello. Yeah, as most of us who work in industry know that this drug discovery takes place in several phases, and the first one, which we do a lot of research, that is called exploratory research, and where we understand the disease in the lab, that is research lab. For example, I did Ph.D., and whatever I was doing was disease understanding, and also sometimes we do target identification. So the goal is to understand the disease mechanism to identify potential drug targets. And during that phase, that is before hit identification, that is discovery phase, that is done in most of the academic labs. So during that phase, we gain a deep understanding of the diseases, molecular pathways, including genetic, biochemical, and environmental factors that contribute to disease progression. And these can involve both literature research and clinical studies. We also use during that phase bioinformatics tools to mine public databases, for example, gene expression data or protein-protein interaction network for insights into the key molecules involved in the disease. So what we get from there? We get to select a target that is involved in identifying proteins, enzymes, receptors, or other biomolecules that play a critical role in the disease's progression. And we find the druggable targets. So what are druggable targets? Most of the targets are not druggable. So we consider whether the identified target is druggable, meaning it can be modulated by small molecules or biologics, for example, enzymes and receptors. And there is also biomarker discovery in that phase. And why do we discover biomarker is because these biomarkers are associated with the disease, and they help define the disease state, monitor therapeutic response, and aid in clinical trial design. For example, in cancer, we might identify mutation in a specific gene that drive tumor growth, making that gene product a potential target for therapeutic intervention. So the first in the list, when we go to the clinical industrial side, we go to the hit identification, and it starts with almost like screening more than 10,000 to 50,000 compounds, and then we come up to after the hit identification phase, we come to 100 to 500 compounds only. So only like 1% of the compounds qualify during this hit identification phase. And this is done through high throughput screening. Also, we call in industrial words, it's HTS, or high throughput screening. It's like a common term that we use in the industry. So during this phase, we develop biochemical or cell-based assays to test potential drug candidates. The assay must reliably measure the interaction between the target and compounds. And common type of assays include binding assays, enzyme inhibition assays, reporter gene assays, and cellular phenotypic assays. There is, as I mentioned, there is a large compound library containing almost like 10,000 to 50,000 compounds that can be screened for potential hits. And these libraries can be of several types. For example, synthetic chemicals, natural products, or fragment libraries. The library size, as I already mentioned, is very large, very big library size is there. And this is usually done by automation, because if we don't use automation, this will take years of time to finish if we do one by one in the lab. So once we have a heat identified, we cannot move ahead. We have to reconfirm that the heat which we generated is correct. So heat confirmation undergoes secondary assays to confirm their activity, and false positives are eliminated at this stage. So we go from 100 to 500 to only 5 to 10 when we go to a lead compound, which we really identify that this is a real hit, and we can go ahead with preclinical testing. So false positives are eliminated during this stage, and hits are validated for specificity and potency. And the goal was to ensure that the hits are specific to the target and have drug-like properties. We also perform target binding validation. That is a special technique called surface plasmon resonance, or SPR, or isothermal titration calorimetry, that is called ITC. Also, we understand off-target screening, because all the hits have to be very selective. So we test the hits against a panel of other unrelated targets to ensure they are selective and do not bind to unintended proteins that could lead to toxicity. We also go for potency testing during this stage, and that determines the half maximal inhibitory concentration. We also call in medical biology terms IC50 or EC50 to evaluate the potency of the hits. So compounds with lower IC50 values are more potent, or we can say that they are better and likely to be effective at lower dosage. So then we have heat optimized. We know that they have efficacy. We go to animal models then, and then this phase is called preclinical testing. And during this phase, as I mentioned, that we have come from 10,000 compounds to 50,000 compounds. So we come to 100 to 50 compounds during heat identification phase. Then we come to 5 to 10 compounds only during lead optimization phase. And during preclinical testing, we use animal models to see the toxicity and other pharmacokinetics and pharmacology assays we perform in animals. And during this phase, 30 to 40% of the compounds fail. Number of preclinical studies include toxicity studies, as I mentioned, pharmacology and pharmacokinetics test, and only a few lead compounds are advanced to these tests. Success rate is almost like roughly 30%. 60% to 70% that in the preclinical development will fail at this stage due to toxicity, mainly the liver toxicity, and which we see during when we take any drug. Most of them have liver toxicity. We complain that liver is not good after taking these medicines. So liver toxicity is one of them. Also insufficient efficacy. So sometimes we complain that the drug is not working, and even after 3 rounds of clinical trials, we complain that drug is not working while the diagnosis was correct. And there are other issues also. So coming to the next round, once preclinical testing has happened, drugs are submitted to FDA mostly, and that is investigational drug, new drug application. We call it as IND application. And then once the FDA approves that, okay, yeah, you can go ahead with the clinical trials. There are 3 phases of clinical trials: phase 1, 2, and 3. And the 4th phase comes in the last, which we hardly have heard about. And that is very important to learn today. So clinical trials, they take almost like 6 to 10 years of time, not in emergency case. For example, during COVID vaccine, it got approved within 1 year, and that was emergency use because we cannot wait for 6 to 10 years for a drug to come to the market. So during phase 1, we have phase 1 clinical trials. During this phase, 10 to 30% of the compounds fail. And during this phase, this is only safety testing. So we take 20 to 100 healthy volunteers. Typically 70% of the 70 to 90% of the candidates move to next stage, because we are not looking for the drug efficacy during this stage. So phase 1, most of the drugs pass. So only like 10 to 30% fail. So in phase 2, we test for the efficacy, and during this phase, 100 to 300 patients with the disease. Now disease phenotype comes here. So during phase 2, 100 to 300 patients with the disease are enrolled, and they are tested for efficacy of the drug candidate. And during this phase, only around 30% of the drugs advance to phase 3. During phase 3, this is a large scale efficacy and safety testing. And during this phase, we enroll around 1,000 to 3,000 patients in multiple centers. So this has to be in different countries, different races, Asian, American, African, everywhere. Whatever is possible. This is the best phase where we see the efficacy of the large scale efficacy and safety testing of any medicine. And about 50% to 60% of the drugs that reach phase 3 eventually get approval. So phase 3, we know that something is in phase 3, and there is, if it has worked on 100 to 300 patients, chances are there that they can work on 1,000 to 3,000 patients also. But there is race issue, there is variation during in different individuals, and that leads to 40 to 50% rejection at this level also. So approximately that 10 to 15% of the drug candidate that enter clinical trials, phase 1, 2, and 3, that only make it through all phases and get approved. And once we have regulatory review or approval, it goes to the market, and phase 4 is ongoing. For example, for COVID vaccine, we have got the vaccine, but we don't know what is long-term effect. What will happen after 10 years? What will happen after 5 years? We got emergency use of the medicine, right? So phase 4 is ongoing, and if there is any problem with the medicine down the line, because the sample size is not in millions, right? We only have 1,000 to 3,000 individual tested in phase 3. It does not tell that what will happen when the sample size is more. When, for example, if it's covering all India, all Japan, all US regions, and the drug that we are taking has severe toxicity. That time, phase 4 clinical trials will be monitored by FDA, and the drug can be retracted from the market. So during this process, all the process, typically takes 10 to 15 years from lab where we work for most of our PhD program. It takes 10 to 15 years to reach to the market with an average cost of $1 billion American dollars to 2.6 billion US dollars. And this varies depending on factors such as drug type, therapeutic area, and developmental pathway. So we started with 10,000 to 50,000 compounds, and we land with only one drug that comes to the market. So you can see how much time does it take, how much effort does it take. So going ahead, so this is like the phases of clinical trials that I discussed already. So some drugs failed that have failed in clinical trial 4 also. So we highly regard companies like Merck, AstraZeneca, Regeneron, Pfizer, but every company's drug have failed in clinical trial 4. For example, Vioxx, manufacturer was Merck, a very well-known company, drug company, and these medicines were used to treat pain, inflammation, mainly due to arthritis and menstrual pain. And the reason for withdrawal was cardiovascular risk. They were causing heart attacks, strokes, and this was discovered during phase 4. And these risks were not observed in earlier trials due to a smaller sample size. And in 2004, the FDA must have told that just withdraw this from the market. And this is like similar to cars manufacturer when they recall their car parts. Sometimes we frequently hear this in US that this body part is not working in your car and there is recall issued. Similarly, in the clinic also, when we have some drug, we see that there are severe risks associated with these drugs and they get retracted from the market. We also have example from Glaxo, SmithKline, we call GSK. In India, we call it Glaxo. It's a very famous brand in the market. And Avendia, that was used to treat type 2 diabetes. They were withdrawn from phase 4 clinical trials, revealed, and the phase 4 clinical trial that was after reaching to the market, that revealed an increased risk of heart attacks and cardiovascular complications. So FDA imposed heavy restrictions, and it was eventually removed from some markets in 2007. Novartis, that is again a famous medicine manufacturer company, and the Gelnorm was the drug which was used to treat irritable bowel syndrome and constipation, and that also caused cardiovascular risk, that was identified post-marketing in phase 4, and this was initially withdrawn in 2007, but then later reapproved in 2019 with restricted use, not for all types of IBS or constipation. So these are some examples of drugs that failed during clinical trial 4, which we hardly care about. We only care about phase 1, 2, and 3. So this is a new thing that many of you must have heard about clinical trial 4. So coming to the next topic, what are the challenges in drug discovery? So common reasons for clinical trial failure and the role of predictive models. So clinical trial failure have several reasons: lack of efficacy, or safety and toxicity issues are there, or they have poor pharmacokinetics and pharmacodynamics. We call that PK and PD. There is patient variability, as I mentioned, that they go to like different, yeah. Oh, sorry. So they go to regions of the world, and patients have variability, and that variability can be genetic, epigenetic, or environmental differences. We also see one reason for failure is inadequate preclinical models. For example, most of the drugs that were tested, they use 2D cell culture system and animal model that often fail to mimic human physiology accurately, and they lead to poor transition to results. Then again, there is cost and resource constraints. These clinical trials are lengthy and expensive. Drug development process that takes years, and clinical trials don't sustain those with marginal benefits. So these are the main reasons of failure of clinical trial. Coming to the to solve these problems. So how we can, is there any question?
No question, sir. संजीवनी कॉलेज नाम से जो ज्वाइन किया, प्लीज म्यूट योर माइक एंड प्लीज टेक केयर कि आपके कारण है ना. प्लीज कंटिन्यू.
So to solve all these issues, there are predictive models which can better predict the outcome of the clinical trials, or before we reach the clinical trials, we have much better data. And one of them is iPSC derived models, which is the main topic of this presentation also. So iPSC derived models provide patient specific insights by mimicking human physiology, and they enable testing of efficacy and toxicity in diverse genetic backgrounds, because iPSC we can make from any individual. That I will come later during my talk. And there is also 3D culture system, mainly as spheroids and organoids, and they better replicate human tissue architecture and microenvironment, unlike 2D system, which I was talking about in the previous slide, where most of the preclinical study previously had used 2D system, but now 3D system is much more in use, and these are called NAMs also, which is like everyday evolving technique in the drug discovery field. So they better replicate human tissue architecture and micro environment, and also improve the accuracy of toxicity and efficacy prediction. There is also in silico models and AI, which is a new field in the drug discovery market, and where people use machine learning to predict drug target interactions and toxicological outcomes. This reduces the need for extensive wet lab experimentation, because we can predict that this is not going to work. We can do docking, and we see that this doesn't dock into a particular enzyme. So this saves lots of time. So in silico models, and AI is very important tool right nowadays. We also use high content screening with advanced tools, and this employs imaging and omics technology to generate data-rich outputs, allowing for a deeper understanding of drug effects. And finally, a very recent and advanced system has come. We call it as organ on a chip, which combines microfluidics and tissue engineering to simulate multiple organ interaction, and this helps to predict systemic effects of drug. So to conclude this, the high failure rate in clinical trials underline the urgent need for innovative predictive models that we also already learned. By integrating iPSCs, 3D cultures, and computational tools, drug discovery can become more efficient, cost-effective, and patient-centric, bridging the gap between preclinical findings and clinical success. So one example that I would like to give is how hepatotoxicity a study has changed. So we were in 2D system, we are going to 3D system, and this is one example, the shift to 3D liver models. So liver damage in drug development is very common, as I mentioned before. 1/3 of clinically used drugs are removed due to liver toxicity, making the liver one of the most commonly affected organs. So FDA requirement is that preclinical toxicity testing, including hepatotoxicity, is primarily conducted in animals before human trials. We cannot go to human. We have to do all the preclinical toxicity study in animals. So what is the limitation of animal model? Can we keep on like sacrificing animals for our use? We cannot. And FDA, US government actually, they are limiting the use of animal, and they told that by 2035 they plan to phase out animal testing. So where is the predictive gap? So approximately 50% of drug candidates cause hepatotoxicity in humans, but not in animals. They are all fine in preclinical stage, preclinical testing stage. They are all fine, but out of those which have passed, 50% of the drug candidates cause hepatotoxicity. So liver toxicity is there reported in humans, as seen with drugs like these so many drugs. For example, we use what we use for painkiller, ibuprofen. It has hepatotoxicity, and we use every day if we have pain in the body. Especially the arthritis patient, they keep on using that, and it has come to the market. But we know that they reach hepatotoxicity, but it's limited, so that it's not getting retracted from the market. So all these, most of the medicines lead to liver failure in humans, but which were retracted from the market, but they had not shown toxicity in animals. So regulatory safety is there that US Environmental Protection Agency plans to phase out animal testing by 2035, highlighting the need for human relevant liver models. So what is human relevant liver model? So current human hepatic models, most of them are 2D, two-dimensional, which we grow in cell, which we do cell culture in the lab, where we grow cells on flat surfaces with media. They are attached to the flasks, and that is called 2D model. But there are better models now that's called 3D, where they are in suspension most of the time, or even if they're not in suspension, they are attached to collagen or different matrices, and that gives them three-dimensional growth property. So commonly used 2D models, for example, primary human hepatocytes or liver cancer cell lines, they often fail to accurately mimic in vivo liver function, leading to potential misinterpretation of toxicology data. However, there are 3D models, as I mentioned, and they better replicate physiological conditions, displaying enhanced sensitivity in detecting hepatotoxicity. So it's clear that we should move towards 3D models now. Coming to 3D liver models, that includes spheroids, organoids, and liver on a chip model. And they allow a spatial interaction between hepatocytes and other cell types, such as parenchymal, non-parenchymal, or extracellular matrix, which we call ECMs, which are critical for mimicking liver maturation and function. So conclusion of all this discussion is there is a urgent need for physiologically relevant models that is called 3D liver models in case of liver. In case of lungs, it will be 3D lung model. It's essential to accurate hepatotoxic screening, improving predictability and safety in drug development. So as we learned about the significance of iPSC and 3D models, before going into details of iPSC, what they are and why we should care about them, we need to learn about a few terms. And the first term is potency. And what is potency of a cell? Potency of a cell is the capacity to differentiate into various cell types, and potency can be of several different types. For example, totipotency, and their differentiation ability is that they can generate all embryonic and extra embryonic cell types. So extra embryonic cell types gives rise to placenta, which most of us know. If we have studied biology, we know that extra embryonic membranes gives rise to placenta. And totipotency example is zygote, when there is fertilization of egg by a sperm, and also the early blastomeres, so the cells from zygote. These are totipotent, means they have the capacity to form the complete body of an animal. Then we go to pluripotent. So pluripotent is everything but extra embryonic cell type. So this is all three genders only, and the examples are embryonic stem cells and induced pluripotent stem cells, which we will discuss in a few minutes. There are also multipotent. Multi means many, so multiple types within a specific lineage. For example, hematopoietic and mesenchymal stem cells. Oligopotent are there, which can give rise to only few cell types within a restricted lineage. For example, lymphoid progenitors, so B lymphocytes and T lymphocytes we can get. And there are finally unipotent. They can only give rise to only one cell type, that is muscle stem cell or keratinocytes. So coming to the ESL based therapy, cell therapy workflow, and if ESL, the one thing to remember is, if ES cells had worked well, there was no need for iPSC to come into to the research field. So I will come to discuss why ESL, what limitations ESL therapy has. So basically, what happens is we collect in vitro fertilized, we collect embryos from IVF centers, where where infertile couples or couples which fail to conceive, they go for in vitro fertilization, and they have surplus amount of embryos which they cannot use, and they usually donate for research purpose. So those embryos are collected, and this was my post MSc training in Dr. Indra Hinduja's clinic in Mumbai, where I was working in a stem cell biology laboratory at NIRRH. It's called National Institute for Research in Reproductive Health. And my training I started with embryonic stem cell biology. And my work was to collect those frozen embryos which were donated by the patients. So these embryos, when we thaw, those are in blastocyst stage. So these blastocyst, so this is like zygote to morula, morula to blastula, then gastula, and during gastulation phase we get blastocyst, and blastocyst has inner cell mass which has the capacity to form, which have the capacity to give rise to embryonic stem cells. So in culture, we see pluripotent embryonic stem cells, means they cannot give rise to all the organs, means they can give rise to three germinal layers, ectoderm, mesoderm, and endoderm, but not the placenta. Placenta comes from this part, which you see. This is called trophoectoderm. So and we are taking cells from inner cell mass only, so they don't have the capacity to form the placenta now. And usual in the lab, when we see ES cell colonies, they form colonies. So if we see a colony, a good colony look like very bright colony, you can see on the top in step number 4. They can be grown in this, and they have the capacity to self-renew. So they keep on renewing, and when we differentiate them, they, as I mentioned, that they have capacity to form all 3 germ layers. And most of our body is made up of or derived from those 3 germ layers. So they can give to liver, muscle, brain, cardiomyocytes, whatever you want to differentiate them into. However, there are limitations of these ESLs. So first one is ethical issue. Many foundations, many organizations, they find that the embryo that we are using for deriving ESLs, they are live, and we are just killing one live human being. That's like we cannot say that no, because they give rise to full grown individual during time, right? If we just transfer those embryos into womb. So this is a valid concern, and that has been taken into consideration by several agencies, government agencies. So this is the major ethical issue, is major concern. Then we also have immunorejection. So even we cannot get all the time like 100% HLA match, and we do get immunorejection because in order to not get rejected, they have to come from the same individual. And we don't know from where those same individual will give rise to human embryonic stem cell, because the embryo is like has gone into the womb and it has developed into a full individual. So that is one issue of immunorejection. Then tumorigenicity is there. So as I mentioned, that these cells can give rise to ectoderm, mesoderm, and endoderm. Even if we use these ESL therapy, and differentiation is not 100% perfect. They can.
प्रोफेसर मोहम्मद इश्तियाक जी, प्लीज कैमरा क्लोज कर लीजिए, कैमरा बंद कर लीजिए. प्रोफेसर मोहम्मद इश्तियाक, प्लीज क्लोज योर कैमरा.
So they will give rise to tumors, and then there is high cost of growing those in the lab. So cost and complexity is another concern that is being considered when we go towards ESL based therapy. And there is severe regulatory barriers. Every country has regulatory barriers for use of these ESLs. So induced pluripotent stem cell in this case provide a non-embryonic alternative. Nobody can challenge that this is coming from a live individual which we are going to kill. No, we are not killing, we are not discarding them. These induced pluripotent stem cells are coming from somatic cells, and they overshadow ESCs in some research areas. So a scientist called Shinya Yamanaka, who is pioneer of iPSC technology, he is a Japanese Nobel Prize-winning stem cell researcher. He was born in 1962 in Japan, and received his medical degree from Kobe University in 1987. His father, he's a doctor, and his father died of hepatitis C. And as a doctor, he could not do anything. He was just seeing his father dying. So this made him to think that what I am doing as a doctor, if I cannot treat my father, and my father died in front of me. So he wanted to be a scientist. He learned that scientists can do something, and he decided that I want to be a scientist, and he started studying stem cells. Most of the disease which we see, they have problem in one type of organs or one type of cells only. So if we can generate those type of cells from embryonic stem cells, is it possible that we can treat those diseases? And this was a motivation for him. So he got a Nobel Prize in 2012 for his pioneering research in induced pluripotent stem cells. So this was the first time that we learned about induced pluripotent stem cell. I will come in the next slide how the field has come up. So he revolutionized regenerative medicine by developing iPSCs and demonstrated that adult cells, skin cells, blood, these can be reprogrammed to become pluripotent stem cells, and they have the capacity like embryonic stem cells. This opened a new possibilities for treating diseases through cell therapy and also transformed the field of stem cell research. So there is no ethical concern now. So is there any question? No. Okay.
No question.
So in 2006,
आपका कैमरा नहीं ऑन हो रहा है ना?
Pardon?
If possible, you can also on your camera.
Oh.
The people will concentrate. नहीं हो
Right.
रहा है. Okay. Okay. Continue.
Okay, so in 2006, so these factors, okay, no. So Yamanaka landmark study demonstrated that a combination of 4 transcription factor can reprogram adult mouse cells into induced pluripotent stem cells. So only we need 4 factors. And 2007, it was shown that human iPSCs can be generated by from a skin fibroblast. So most of the skin fibroblast comes from foreskin from the penis. Those are the cells which are easily available, and that paves the way for patient-specific stem cell research and therapeutic applications. In 2008, it was first demonstrated that iPSC-based disease modeling can be done, showing the ability to recapitulate the pathological phenotype of a genetic disorder in vitro. And in 2010, there was development of integration-free method. So most of the previous method used integration-based method for these 4 transcription factor, which he used in 2006 and 2007. In 2010, there was integration-free method for generating iPSCs, reducing the risk of genomic alterations, because if we do integration, there is genomic abnormality, there is alteration, and that improved the safety of the technology. In 2012, there was first clinical trial using iPSC derived cells for the treatment of age-related macular degeneration, a leading cause of blindness. And in 2012 only, Yamanaka got Nobel Prize. In 2014, generation of iPSCs from peripheral blood cells, which is very easy to get, not for skin now. Now we can get from the blood, and that enabled a more accessible and less invasive source of patient-specific cells. And in 2016, the use of iPSC-derived cells in regenerative medicine, including the successful transplantation of iPSC-derived retinal pigment epithelial cells in patients with macular degeneration. So this started macular degeneration project started in Japan, and she was a very good friend of Yamanaka, and we call now all the those 4 factors as Yamanaka factors. So by now, all of you must have learned that how iPSC holds the promise for drug discovery, because we treat them as ESL only, having all the property of ESL, and ESLs can give rise to all the cell types you can see in a step 5, right? All the cells which are derived from ectoderm, mesoderm, and endoderm. So here is one example of use of patient-derived induced pluripotent stem cells for neurological disorders. So we have patient somatic cells, we need to reprogram them. So somatic cells can be coming from blood or skin. So we reprogram using 4 factors, which are called Yamanaka factors. These are OCT3 or 4, mostly OCT4 in 90% of the case. OCT4 are used. SOX2, KLF4, and CMYC. These are transcription factors, and they give rise to patient-derived iPSCs. Now we have whatever we want, whomever we want to treat, we are getting cells from those individuals only. We are not like generating a common platform here of iPSC. We are going towards a patient sample. So we have patient-derived iPSC, and we can differentiate them into neurons. So these neurons will have mutation. If there are mutated neurons, mutation is present in the patient, we will have mutated neurons differentiated also. But at the same time, what we can do is we can use CRISPR-Cas9 to correct those mutations. So CRISPR-Cas9, somebody on this platform, people have at least I think 3 different scientists have presented about CRISPR-Cas9, how gene editing. So this is a method of gene editing where we can correct the mutations. We can add some extra DNA or remove some extra DNA. Whatever we want to do, we can do using CRISPR-Cas9, which is very targeted approach, and we get genetically corrected iPSCs, and then we can differentiate and get corrected neurons. So this is called isogenic control. This is a control. We have mutation one, but we also have the corrected one, and this is called isogenic control. So now we also have age-matched healthy donor somatic cells. So 56 years, for example, one patient is 56 years old. We will get a healthy individual of the same race, not from, like, if I'm from India, I cannot get someone from US, and white American. No, we cannot do that. We should go to age-matched, race-matched individual, and that can serve as a healthy donors of the somatic cells, and we can reprogram those cells, and these are just a control. Okay, so in science we always have control, positive control, negative control. We are having generating control here. So we will reprogram using again Yamanaka factors of 3 or 4, SOX2, KLF4, and CMYC. These are transcription factors, and then we'll have control derived iPSCs, and as I mentioned, we can differentiate them, and these will give rise to healthy neurons. This is again a control. So we have control, healthy control, we have isogenic control, and we also have mutation carrying neurons from individual. We are going towards drug discovery. Okay, so then what we will do is we will identify the disease phenotype. So we have patient neuron coming from mutation. We have 2 types of control. One is healthy control, one is isogenic control, which is corrected neurons. And then we perform several different studies like electrophysiology, immunocytochemistry, RNA sequencing, bulk RNA sequencing, single cell RNA sequencing. So RNA sequencing will give us RNA profile, messenger RNA profile, basically. Immunocytochemistry will give us protein profile, and electrophysiology will give us how the signals are going through those neurons. And then we will use high throughput screening on these. So now the drug development process comes immediately. So we have different drugs tested, and we will see that which one is effective in controlling cellular physiology or disease phenotype, and this gives rise to drug discovery, and we test in vivo safety and efficacy. So the final one, we are moving towards the last part of the presentation. So towards this, we are going towards the organoid system, that is 3D model. So we have somatic cells, we reprogram, we get cultured iPSCs, and then we differentiate in the form of organoids. Organoid is 3-dimensional assembly of a stem cell having a property having cells. So if there is no stemness in those cells, that will give rise to a spheroid, not organoids. So for organoids, one thing to remember is they should have a stem cell property, and then we will have cell phenotype analysis. We will do disease modeling, perform disease modeling and therapy, and this will give rise to personalized therapy, which medicine is working for what type of patient, and we will have these patient with neurodegenerative disorder treated. So these are the advantage and limitations of cellular models which I have discussed. So monolayer culture, which most of the 2D, this is also called 2D culture, which most of the labs were using before. They have high throughput, means you can do lots of screening using automation. And then we have a spheroid cells which do not have a stemness, not a stem cell property. Organoids, which are three-dimensional cells, again like spheroids, but they have a stem cell property. 2D culture is low cost, easy to obtain. Every labs everywhere in the world they grow 2D cells. Simple to culture. Anyone, even a 10th grade student, if we teach them how to grow cells, they can grow. And fast preparation time is there. But the limitation is they have only single cell type, no cell to cell interaction. There is no cell to extracellular matrix interaction. Limited culture time, we cannot grow for unlimited time, and incomplete metabolic function is there. For aspheroid and organoid, most of them mimic physiological architecture. Organoid is much better in terms of mimicking physiological architecture. They replicate cellular functions, and they can be long term, they can be grown long term in culture, and they also have multi-cell type interaction. We also have organ on chip. Here it's example of a liver on a chip, which I discussed already in previous slide. However, the disadvantage of this system is there is lot of variability in preparation. Every lab has different approach to prepare these spheroids and organoids, and they take a long time to generate, especially organoids. Spheroids are easy, organoids are a little bit expensive, time consuming, and laborsome. So there is high data variability because it depends on what kind of cells are they really having a stemness property or not. That matters a lot in organoid, and it lab to lab variation is much more. So this is one challenge that we have in organoid and spheroid system. So iPSC based therapies are now coming up, and one of the disease is age-related macular degeneration. This is called retinitis pigmentosa that I discussed already in the previous slide about this condition. And the approach was iPSC derived retinal pigment epithelia cells, and clinical trials are ongoing in Japan using these cells to treat AMD, and the goal is to evaluate safety and efficacy of implanting these cells into patient eyes. And she is the scientist Masayo Takahashi from Riken. Then we have another clinical trial going on in monkeys, that is for Parkinson disease by Dr. Jun Takahashi and his husband of Masayo Takahashi. So both husband and wife are in the clinical trial phase, and that's a proud moment for the family too and for the entire Japan. Then recently, Fate Therapeutics, they received the FDA approval for iPSC trial to treat solid tumors with an off-the-shelf natural killer cell cancer immunotherapy. And another one therapeutic company is called Health Therapeutics. They received FDA approval for IND. So IND is after the preclinical study. So they got IND application for iPSC derived heart failure cell therapy. So hopefully we will have in next 5 to 10 years all these in the market. So these are the limitation of, so we have promised for iPSC. There are always limitations of any technique. So iPSC also has limitations similar to ESL, embryonic stem cell. In iPSC, we have tumorigenicity because they can also give rise to all three germinal layer cells. So if even like 10% or 5% cells are not differentiated into particular lineages, they will give rise to tumor cells. We also have genetic instability. As I mentioned, that there was integration based method to prepare these cells. So genetic instability was an issue. Limited differentiation efficiency in some of the cases. There is also immune rejection. Even if we have very foolproof iPSC differentiated and from the same individual, there are chances of immune rejection in some cases. There is high cost and scalability issue. Long-term functionality is still a problem, and there are ethical and regulatory concerns which people don't know still that what will happen. Every case is a new case for regulatory bodies. In the similar thing that I discussed is for organoids. There is lab to lab variability, and it's an upcoming field in the drug development area. They sometimes they don't so mature organs. For example, if we need to get mature neurons, there may be immature neurons 90% of the 90% of the time, and 10% only is mature neurons. So maturity is an issue. Complexity and cost. These organoids are very complex, and it's expensive to grow them. Limited integration, and also there is no vascularization, so we do not have blood flowing in them. So there is limited oxygen supply to all the cells which are present in organoid system. So this is final slide, and this is regarding future outlook and opportunities. So using these iPSC and 3D culture model, we can have accelerated drug discovery. We can have enhanced drug predictive accuracy. We can reduce animal testing, so that is a very good sign, I think. And we will have personalized medicine potential using them. So with this, I would like to thank all my colleagues, my supervisors, Professor M.R.S. Rao, Dr. Jeff Hayes, Art Scolci, and Anthony Gagliardi, who have taught me culturing cells, even Deepa Bhartiya and Indra Hinduja, who trained me initially in the embryonic stem cell biology. And thank you all for coming today and listening, and I hope you learned something today.